Hello, everybody. My name is Bogdan Parakhonsky. I'm a researcher, postdoctoral researcher from Ghent University. And today we will talk about the development and construction of biocarriers based on the mineralized hydrogels. Why we need really the biocarrier? What it is about? It is a carrier. It is a kind of composite materials which could contain a few different components, like the minerals, hydrogel, biomolecules, and most important part, the kind of living organism, like cells or bacteria. And such combination could provide additional function of this biocarrier, like the delivery function, detection function, self-assembling function, kind of construction materials, and others. There are two main types of the possible biocarriers. One of them is kind of flat films, which functionalize with kind of molecules or bacteria or cells. And second one is biocolloids. In my presentation, I will talk about the one example of the biocarrier formation and application for tissue engineering and drug delivery. For example, when we start creating the biomaterials, for bone tissue regeneration, we like to create new materials which has a lot of cells inside and has as much cell as possible, as much minerals as possible, and as much functional molecules as possible. And for this reason, we need kind of biocarrier. As a component of biocarrier, we decide to use a kind of mineral biomolecules which can provide the matrix and kind of additional function. The hydrogel is a matrix which glue all components together, and cell and bacteria is a functional living organism which will be support the bone regeneration. There are two different approaches how to synthesize the materials with such minerals. In our work, we choose minerals like the calcium carbonate because it's one of the main source of the calcium for bone regeneration. And as a result, we can create the colloidal particles of these materials and start creating gels around these particles or add these particles to the gels. It's one approach. The second approach, it is a mineralization or synthesis of the calcium carbonate in the kind of scaffolds or inside the gels. Yeah. First of all, how to synthesize calcium carbonate particles and what it is. Usually calcium carbonate exists in three modification, like the aragonite, calcite, and vaterite, but really in our work we are mostly interested in the vaterite phase, which are porous polycrystals. And really, if we manipulate with different conditions, like the time and temperature, we can create different size and shapes, and could use these particles in the following works. Really, how we apply these two approaches? Respect to the biomaterials for bone tissue regeneration. We synthesize our particles, which method I described before, and we can add them to the gels, or we can apply this method of the synthesis particles inside the gels and create the bone implants. Both of this approach make sense for their own application. One of them, it will be self-gelation to fill kind of holes in the bone. Another one, it will be bone implant coating, which could enhance the cell adhesion. But good or not, we will try in our work. First part, it will be cells in gel. For this reason, we can have a gels, and we can premix this gel with the cells and add the particles in it. And we can see that depends on particles composition, the cells start to be grows much more better than without particles, and then provide them more better grows the hydroxyapatite particles, which are important for the bone regeneration, which is exactly part of the bones. In the case, depends on our parameters of our system, depends how much particles, how much colloids we add. It will be strongly influenced to the final gel property, how the cells grows inside the gel and how they functionalize. Second part, it is how to grow the cells on top of the gel, like for coating the bone implants. For this reason, we apply two approaches. We take a gel which usually cell not grows well, and we try to concentrate them and dry to enhance the mechanical property and mineralize them. And depends on these two different approaches, we have 4 different samples, like the gels and dry gels and mineralized and not mineralized. And what we get as a result that the cells much more better grows if it's mineralized dry gel. And we try to understand why it's happened in such that the cells grows well just in these materials which can contain all our possible components, like the cells and minerals and the gel together. And we do very complex analysis, which involves the atomic force microscopy technique, cryoscanning electron microscopy technique, the confocal microscopy technique, and realize that the cells, as a kind of living objects, like to attach to the hard centers which provide by the minerals inside the gels. It means that the particles quite floating in the gel matrix, and the cell attached to this hard center in it. And the case grows much more better. And as a result, they provide better apatite formation and better bone regeneration. The potential on such approaches could be in wound healing, in bone implants, in different coating, and as well, if we go further for this kind of biocarrier applications, could be in 3D bioprinting. Because if we can formate materials which can provide nice cell growth, we can formate them more complex tissue. General message of this presentation, if we use our object as kind of building blocks, we can combine them in different way and can create different materials with completely different property, like the hydrogel with particles, hydrogel on particles, particles could be inside the cells, outside the cells, and all of them has own applications. Yeah, thank you very much for your attention.